Good morning, everyone. I'm Steve,
And I'm Scott.
and together we're Backyard Musings in the Backyard, Apple Valley.
Back in the backyard, but don't get too used to it. But today we're talking about a critter that is not too many people's favorite, that's for sure.
The ants go marching on, right? Ants rely on an extraordinary sense of smell to build and organize their societies. Each of their neurons can detect only one specific scent, a rule that keeps their chemical messages clear. Scientists studying the clonal radar ant have now discovered how this works. Each neuron activates one odor gene while silencing all the others. Sounds simple, right? The finding reveals a hidden genetic process that helps ants communicate, survive, and evolve their complex social systems. Would you like to know more?
Ant societies rely on scent to function. Pheromones,
Pheromones.
pheromones help them find food, alert each other to danger, and coordinate the daily life of their colonies. This chemical language depends on a simple biological rule: one receptor per neuron. Inside an ant's genome are hundreds of odorant receptor genes, each responsible for detecting specific smells. If a single neuron were to activate more than one receptor, the signals reaching the brain would overlap and blur, destroying the ant's precise sense of smell. How do they come up
Makes
with this
sense.
stuff? How do
Makes
they come
sense.
up with this?
Well, they got to dissect the ant. Autopsies.
Okay,
Ant autopsies.
gotcha. Researchers studying the clonal radar ant have now uncovered how each neuron chooses just one receptor from hundreds of options. Their work, published in Current Biology on September 19th, 2025, resolves a long-standing question about how ants preserve clarity in their chemical communication system.
Long-standing question.
Okay.
People want to know. Quote, "We're describing a new form of gene regulation," says Daniel Cronauer, head of the Laboratory of Social Evolution and Behavior at Rockefeller. So, social evolution and behavior of ants or humans or dogs or mammals?
I don't know.
There's a lab that like that? Okay.
Apparently so.
Our results demonstrate the importance of studying less conventional model species. We were able to discover new, fundamental molecular phenomena in clonal radar ants that we could not have seen in fruit flies.
Okay, what a comparison.
Fruit flies are tiny too.
Yeah. The key rule in the science of smell is that every sensory neuron must have its own molecular identity.
Of course.
Quote. Yep, here we go. It's a kind of dogma in the field of sensory neuroscience, says Giacomo Glotzer, a graduate student in the Cronauer lab. Another quote: each sensory neuron typically expresses one receptor, and that gives it its identity.
Sounds simple. Different organisms achieve this one-to-one pairing in distinct ways. In fruit flies, which we mentioned earlier, molecular switches precisely turn individual genes on or off so that only one receptor is expressed per neuron. Mammals, like us, use a more unpredictable system in which neurons randomly reorganize their chromatin until just one receptor gene remains active.
Until now, it was unclear whether ants followed a method similar to flies or mammals, or something entirely their own. Unlike fruit flies, which have about 60 receptor genes, ants possess several hundred, numbers closer to those of mammals. Okay, I didn't even know we had them. Many of these genes are grouped into clusters of nearly identical sequences, making it difficult to activate one without unintentionally triggering others. Such genetic crowding means the straightforward approach used by fruit flies would not work for ants, implying that these social insects have evolved a unique way to preserve their, I don't know why that's in quotes here. Is that you? Did you have that? Or
No,
okay.
that's
One receptor, one neuron balance.
So I was just thinking while you were saying this, Scott, is there any doubt now that there is a supreme creator that designed all of this? I mean, they didn't walk out of the water and develop this as ants.
Why? My question would be, why do we even have ants? What do we have? They do not live long in my sphere of influence.
Are you an ant hater?
We do not like ants. Yeah,
Huh.
we do not. Now, ants outside are fine, like the what do you call them? Carpenter ants or worker ants or whatever.
You don't like fire ants?
The little red grease ants and stuff that come in your house and destroy stuff. Yeah, like those don't last long. Taro, and they're not a sponsor. Maybe they should be though. Taro. Have you ever had ants problems in your house? Taro.
Yeah.
Terro. It's a borax type liquid. You put it down and they swarm on that stuff. Take it back to the nest. Done. 2 or 3 days. Done.
Yeah, I didn't know that.
Yeah, it's good stuff.
But I mean, this points to this is so complex.
Oh yeah, back to
the genomes
topic.
And stuff. This Heavenly Father created this.
Yeah.
All of these animals that have these complex stuff. The
genomes,
flies. The DNA, even fruit flies, even what are those little pricker things out here? The
goat heads.
And the goat's heads. That's probably Lucifer probably came up with that one. The goat's heads.
Well, it's still around.
Yeah. Okay. Building on a foundational paper on the subject that the team had published in 2023, the lab set out to capture this elusive mechanism in action. After dissecting the antennal tissue of clonal radar ants, they probably had a microscope for that, like a big one. The team then used RNA sequencing to determine which genes were turned on and RNA fluorescence in situ hybridization to localize those genes in the ant antenna. Then they used numerous cutting-edge molecular and computational techniques, AI, to create a clear image of one chosen receptor surrounded by its quieted neighbors.
They found that when an ant neuron switches on its chosen receptor gene, it doesn't stop there. The RNA polymerase, the engine that copies the DNA into RNA, continues past that gene's normal endpoint, spilling into the genes that sit downstream of the target. These read-through transcripts remain trapped in the nucleus, likely because they lack the unique tag required for export. The authors speculate that these transcripts are non-functional, but that their production itself is what silences downstream genes. Meanwhile, the neuron also generates antisense RNAs in other direction. The polymerase here acts as a roadblock to silence upstream genes that might otherwise have turned on.
That's all fascinating.
Yeah, well, I guess.
Probably years of
way
study
over
too.
my head.
The result is a protective genetic shield around the chosen receptor gene. I can understand that. Quote, when we took the mechanism apart and dissected it into its constituent parts, we found that this strategy serves to silence the local genomic environment, giving that cell its singular receptor identity, says Parviz Daniel Hijazi Pastor. That's the name. Pastor, that's the name.
Okay.
Parviz Daniel Hijazi Pastor, a biomedical fellow in the clonal lab. Quote, our findings center around transcriptional interference, that the neuron chooses one receptor by preventing the true transcription of other receptors, both upstream and downstream.
The team went on to confirm that the same mechanism is at work in other social insects, including the Indian jumping ant
I don't even want to think about
and
that.
the honeybee. Oh, okay. These findings raise the possibility that many insects, both social and non-social, use transcriptional interference to maintain a 1-to-1 ratio between receptors and neurons.
The implications extend far beyond insect olfaction by showing that tight clusters of related genes can be governed by two-way safeguards, read-through that requires downstream neighbors, and antisense transcription that blocks upstream ones. This work offers a blueprint for how genomes might keep large gene families in check. The results also point to a potential mechanism for explaining how ants quickly expand their sense of smell over relatively short evolutionary time.
The findings described in this paper might allow newly duplicated receptor genes to be integrated into a sensory system without the need to co-evolve additional regulatory mechanisms. Okay. Did you ever watch the show Twilight Zone?
Oh, yes.
And there was another one that was like the Twilight Zone. I can't remember
there's
the name of that.
a short-lived one. Yeah, I know what you're talking about.
So, did you ever see the episode where those giant ants?
Outer Limits.
Outer Limits. I think it was either one of these shows, but they had those giant ants, and they had human faces on them, and they were taking over everything. They were everywhere. Oh, I'm telling you, I watched that.
Did
thing.
they speak
I was
English
terrified.
or Russian?
I'm getting chills just thinking about it. No, I don't think they spoke.
Black and white. It was black and white.
It was black and white. Yeah.
Fascinating stories back then.
They were all over the place. They were crawling on cars and just taking over.
That was the original scary,
yeah,
stuff back then. What is it? Early 60s, late 50s?
60s, yeah, 60s, I think. Yeah, what was that guy's name? You have entered the Twilight Zone.
Yeah, Rod Serling.
Rod Serling, yeah.
There'd never be another one like him. He was just unique,
Yeah.
Howard Cosell, unique.
Yeah.
All right, folks. Thanks for taking a walk down memory lane with us.
See you this afternoon. Take care.